Hello, everyone. Please excuse my voice. It's the worst time to have a cold, but please bear with me. My name is Pardis. I'm a PhD student in Dr. Blair Levitt's lab. Unfortunately, he wasn't able to make it. He was the one invited to give this talk. So I really appreciate the team to give me this opportunity to come and speak on his behalf and present some of the work that I've done on GNAO1. So the title of the project is Developing a CRISPR-mediated gene therapy for GNAO1 encephalopathy. And we are based in Vancouver, University of British Columbia, and this is our beautiful campus. I have a very brief introductory slide. I know we've all seen different versions of this, so I will go through it very quickly. The GNAO1 gene codes for G-alpha-O, which is a subunit of a heterotrimeric G protein complex called GO. And once it's activated, it binds GTP and dissociates from the beta-gamma subunits. And both of the units will have their downstream actions, including indirectly inhibiting adenylyl cyclase for cyclic AMP production, inducing calcium channels, inducing potassium channels, inhibiting calcium channels and neurotransmitter release. And all of this will result in neurotransmitter release differences, neural developmental phenotypes, synaptic plasticity, excitability, movement function, and also epilepsy. So in this project, we work with R209H variants. Initially, we started the project with one patient that we received stem cell lines from, a local Canadian patient who harbors the R209H mutation. And then through collaborative work with Dr. Harold Micker's lab, we were able to get a second iPSC line harboring the same mutation. And these were all through facilitations by the Bow Foundation. So the project is divided into 3 different aims, and I will briefly go through these aims and introduce them, and then get into details of the experiments that I've done for the aims. So the first aim is to develop and optimize a CRISPR genome editing approach for targeting GNAO1 exon 6, which is where R209H is located. And then this is done through screening different DNA repair templates, guide RNAs, CRISPR-Cas9 systems, doses, in a relatively easy to transfect cell lines for high throughput screening, such as HEC293 cells. And then in aim 2, our optimized parameters will be carried forward in more therapeutically relevant cell lines, such as the patient stem cells, as well as primary cortical neurons generated from the R209H mice that we received from Dr. Newbig's lab. And then lastly, aim 3 is preclinical testing in a GNAO1 mouse model of R209H. So the overall hypothesis for the project is that correcting R209H in patient iPSC-derived neurons and mice using this approach will significantly alleviate cellular pathology and movement disorder-associated phenotypes. So brief intro to CRISPR-Cas9 as a genome editing tool. It's composed of a guide RNA, which is an RNA molecule bound to the Cas9 protein, which through sequence complementarity will find a specific target in the DNA and make, where the Cas9 nuclease will make a double-stranded break into DNA. And in the presence of a DNA repair template, homology-directed repair will introduce a desired sequence of DNA into where the guide RNA was designed to target, and repair the DNA in the sequence that we desire. So effective CRISPR-Cas9 editing really depends on the way we deliver the machinery into the cells. There are multiple platforms to deliver CRISPR-Cas9. Viruses are very popular, but now lipid nanoparticles as a non-viral delivery platform are becoming very popular, especially after the COVID-19 vaccines that had the same type of lipid nanoparticle formulation to deliver nucleic acid. So they've already been FDA approved and and shown clinical promise in that sense. Mainly, there are 4 components in lipid nanoparticle formulations, although there are more components added for specific targeting or delivery of specific different cargoes. There is ionizable cationic lipids as the core, phospholipids, PEG lipids, and also cholesterol. So the specific lipid nanoparticle formulation that we use is somewhat similar to the COVID-19 vaccines, although it's specifically formulated to deliver CRISPR in the ribonucleoprotein form, which is a Cas9 protein pre-bound to the guide RNA. So this has advantages because it's bypassing the delivery of DNA as a plasmid and also mRNA of the Cas9 by delivering the preformed guide CRISPR-Cas9 machinery. So the mRNA of the Cas9 that has to be transcribed in the cytoplasm has to be in the right concentration and timing for it to bind the guide RNA, but this is bypassing all of those steps. And also it has reduced off-target effects as the Cas9 protein is able to be cleared faster from the systems. And this formulation is called Incisive DS, which is a formulation that was developed in the Levitt lab that is now being developed for different therapeutics at Incisive Genetics, which is a company founded by my supervisor, Blair. So how do these lipid nanoparticles enter the cells? They are mainly endocytosed via the low-density lipoprotein receptor pathways. So the LDL receptor, which in this microscopy image is the green specks on the surface of the cells, will bind lipoproteins on the corona of the lipid nanoparticles. And then it's endocytosed into the cells, where the RMP molecule will then enter the nucleus using a nuclear localization signal that's embedded within the CRISPR-Cas9 protein, and then go on to do the gene editing. So I will now get into more details of the aims. The first aim was to develop the system in a relatively easy to transfect cell line like HEC293 cells. So the first thing we wanted to look is, are we able to target GNAO1 as a whole gene with this LNP-RNP system? So we designed multiple guide RNAs to target multiple exons to maximize our targeting capacity, and then measured DNA editing via mRNA reduction by qPCR of guides on their own and in combination with one another. So we see that in this case, guide RNA 356 in combination results in the most efficient gene editing that was measured with mRNA knockdown by qPCR. So the next step would be to design guide RNAs specific to the locus where R29H mutation resides, which is exon 6. So additional guide RNAs were designed, 7, 8, and 9, with 5 that was also in close proximity to the mutation, so it was appropriate to screen for HDR with this guide RNA as well. And this is still in HEC293 cells, so the DNA repair template in this case carries the mutation R29H, where we sequenced for the presence of the mutation to assess our HDR efficiency. So the RT-PCR is the qPCR mRNA reduction graph again. This is all one experiment, just divided into different graphs. So we see that in this case, guide RNA 5 also is the most efficient guide that results in the highest percentage editing, which is just total number of indels, and then also the highest number, around 10% of HDR when done Sanger sequencing. And here, luciferase guide RNA is used as a negative transfection control. So we tested our ability to target the GNAO1 gene as a whole, and the next would be to move on to more therapeutically relevant cell lines, which is the patient stem cells as well as primary neurons generated from the mice. When we first received the patient stem cells, they were newly generated and not characterized. So just based on standard protocols, we characterized them for pluripotency markers through staining, and also looked at their trilineage differentiation capacity by differentiating them into the endoderm, mesoderm, and ectoderm lineages, and used qPCR with 2 different genes or markers from each lineage, and this was all done with a darker blue bar graph of a healthy control stem cell line, and DNA sequencing to confirm the presence of the mutation in the patient stem cell lines. We're also doing karyotyping, although the data is not shown, but that's also going to be added in our characterization. Another next step of characterization of the patient stem cells we did was neural stem cell rosette formation, which Dr. Meekers also talked about. So this is an assay to mimic neurodevelopment in the stem cell lines. And although statistically not significant when quantified, we do see qualitatively that the neural rosettes formed in the patient's stem cell lines are variable in their embroid body sizes and have aberrant shapes. And similar to what we saw earlier with the previous talks, the neural rosettes also lack the wild-type polarity that we see. And the next would be to generate primary cortical neurons from the GNAO1 mice that harbor the R29H mutation. So the protocol we use in the lab is set up timed pregnancy for the mice, where after a very specific date based on what type of neurons we want, 17.5 days of pregnancy, we dissect the pups from the uterus and dissect the brains out and culture the cortex only to culture primary neurons. And characterizations of these cultures are shown here in this microscopy images. The left is a GNAO1 marker green and DARP32, which is a striatal marker. In this protocol specifically, the striatum is dissected out, so it's a control for looking at how efficient or clean the dissections are. The middle image is GNAO1 and IBA1 for controlling for microglial contamination. As you can see, there is one microglia in the middle here. Some of them do make their way. It's impossible to get 100% pure neuronal cultures, but as long as they are at an acceptable low percentage. And also the last one is astrocytes. So you can see these astrocytes that also are present in the culture, but again at very low percentages. So these are the characterizations of the primary neural cultures. So next would be to choose an appropriate cellular outcome measure for analyzing our therapeutics benefit post-CRISPR-Cas9 collection. And based off all of the work that the scientists here have done, we have picked two assays that are commonly popping up for GNAO1 iPSCs, which is neurite outgrowth and also calcium-dependent signaling, although we are also considering MEA analysis, but since we haven't established the assay, it's not included in this presentation yet. So I have some data to show on the neurite outgrowth analysis. In order to do the neurite outgrowth, we have to generate cortical neurons from our iPSCs, and this is a monolayer differentiation protocol, unlike EB, embryo bodies, which is 3D differentiation. So on day 0, we take the iPSCs and introduce them to a neural induction media. For about 7 days, we do media changes, and these will give us a culture of neural progenitor, or MPC cells, which we then culture for about 3 to 5 passages to establish a stable and more homogeneous MPC culture in an MPC expansion media. And then we can, at this point, freeze down the cells and bring them up later. So there's that advantage of using this MPC protocol. And then we culture the MPCs in a neuronal differentiation media for the remaining of the protocol in order to get mature cortical neurons, which is established on day 43. And it's at this point that we have done our neurite outgrowth assay. So the outgrowth assay we use is a live cell imaging to assess the neurite outgrowth of the cells. The machine instrument, sorry, I'm not sure why it's low quality. The machine we use is called IncuCyte, live cell imaging analysis. You plate the neurons on day 42, you passage them or dissociate them from the plate them in a new plate, and then insert it into the machine. It's a live cell imaging, so it keeps track of the outgrowth of the neurons. It's a little bit hard to see, but the yellow linings is the machine recognizing the cellular bodies, and the purple is the axonal outgrowths that are being traced by the machine. And then you can use this data to graph the rate of the outgrowth of the neurites, and you can compare them across different genotypes. So the different cell lines that we did this assay with was the first GNAO1 patient, local Canadian, that we got, and the second one that we got from the Meekers lab. and it's isogenic line against a healthy control line on day 43 of the neurons. So this is the result here. The first patient that we have is dark blue, second is the light blue, and the pink is the isogenic of the light blue, and the purple, and the pink one is a wild type. So we see that the GNAO1 R209H harboring neurons exhibit a more rapid neurite outgrowth. And this is shown by the curves in the graph over a 48-hour assessment. We do see that since it's still exponentially growing, we can extend this assay even further, maybe to 72 hours, to establish a more robust difference between the patient lines. And we see that the patient 2 and its repaired cell line has a significant difference in the rate at which the neurites are growing over 48 hours. And these are some representative images of each of the lines of the graph that I just showed you. So the first patient line is here. This is after a little bit after 48 hours at the end conclusion time. Patient 2, isogenic of the patient 2 and the wild type, and we see that how the wild type and the repaired are more similar to one another than the ones carrying the R209H mutation. So the next step would be to screen guide RNAs for CRISPR-Cas9 homology-directed repair of the mutation with the sequence of the patient lines. So far we've been doing it in hex, but these are in the MPCs generated from the first patient line. So we designed even more guide RNAs to screen, numbered from 7 to 13, with 2 different DNA repair templates based on where the PAM site is cutting on the DNA. And then on this graph, percent editing is each of the guides, where the lighter blue is the percentage of indels, so overall gene targeting, and the darker blue is homology-directed repair, so the correction of the mutation in this case. And then from this result, we see that guide RNA 7 is the most efficient one with the least variability between the samples as well. MPCs and hex grow a little bit differently, so that could explain why we see some variability in these lines, but out of these guide RNAs, number 7 is the most efficient one. So the next steps here would be to enhance this even further. 10% might seem a bit modest, but there are many hurdles that this CRISPR-Cas9 machinery is going through. So for a first proof of concept, I think it's actually not bad. So the LNPs have to encapsulate the Cas9 efficiently. The guide RNA has to find the DNA sequence efficiently. And then the DNA repair template has to be present at the right place and right time for homology-directed repair. So 10% for all of that is not bad for the first try, I think. So I want to also mention here that these 2 guide RNAs, 7 and 11, the pink ones, are also placed or designed to be placed on exon 6 for base editing as well. So what we have also tried, and this is a current experiment that's ongoing, so I don't have the sequencing result yet, but the transfections have been done, is to use these 2 guide RNAs to compare the efficiency of Cas9 versus a base editor, ABE8E protein. So the nature of the R209H mutation, which is a conversion of an A to a G, is kind of a low-hanging fruit for a base editor like ABE8. Because it converts G back to an A. So we also wanted to try to see if we can get more efficient gene editing using a base editor. And the next step after this would be to do an LNP-RNP correction of the most efficient form of endonuclease, a Cas9 or ABE8, and then look at the assessment, and then assess its effect on the neurite outgrowth assay, and then the calcium imaging that I talked about. As the last part of the project is preclinical testing in the mice. And the mice we received are from Michigan State University, Dr. Newbig's lab. They're the R29H mice, and their most robust phenotype is in open field, where they show hyperactivity. So our very simplified workflow for this step would be to do intrastriatal direct injections, so within the stratum of the brain, of the candidate CRISPR-Cas9 drug. And then first do a short-term biomarker-based analysis, such as tight sequencing, which is looking at the percentage of gene editing, to see if we're able to get efficient correction of the mutation first. And then the second would be to do a long-term preclinical efficacy analysis, which is to look at behavioral phenotypes such as open field and some of the gait abnormalities that was mentioned in the paper. And this will be assessed at multiple points over a span of 6 months. And then at the conclusion of the experiments, the brain will also be collected for sequencing to correlate behavioral phenotypic correction into the percentage of gene editing. So with that, a summary of the work so far is that we were able to target GNAO1 using LNP-RNP in an HEC293 systems. And a candidate guide RNA was chosen for HDR correction of R29H through screening of multiple guide RNAs. And the initial patient iPSCs that we received have been characterized. And we were able to add another characterized patient iPSC line harboring the same mutation and also its isogenic line to our project. And also a neuronal differentiation protocol and a relevant therapeutic outcome measure, which was a neuroid outgrowth assessment I showed, has been established. And then we will be testing a Cas9 versus a base editing correction for R29H. With that, I want to acknowledge my supervisor for supporting me through all of these projects and our collaborators at the Pulati Lab who help us with the iPSCs. And of course, the Bow Foundation, Dr. Newbeck and Dr. Mickers for providing us with the stem cell line and the mouse model.